Imagine a future where a failing organ no longer means a life-threatening wait for a donor. Picture a world where new kidneys, hearts, or livers can be custom-made on demand for those who need them most. This vision, once confined to science fiction, is rapidly becoming a tangible reality through the groundbreaking field of 3D printing human organs. This revolutionary technology promises to transform medicine as we know it. It offers a beacon of hope for millions suffering from organ failure worldwide. We're on the cusp of an era where biology meets engineering in the most profound way. The global organ shortage is a dire crisis. Thousands of lives are lost each year due to the scarcity of suitable donor organs. Current transplant methods, while life-saving, are limited by availability, immune rejection, and the need for lifelong immunosuppression. Traditional treatments like dialysis for kidney failure or mechanical pumps for heart conditions are often temporary solutions. They significantly impact a patient's quality of life and do not address the root cause of organ dysfunction. A new paradigm is desperately needed. At its core, 3D bioprinting is an additive manufacturing process. It involves depositing layers of biological materials, known as bioinks, to create three-dimensional structures. These bioinks are not just inert substances, they are carefully formulated mixtures of living cells, growth factors, and biocompatible polymers. The process precisely places these cellular building blocks according to a digital blueprint. This blueprint is often derived from medical imaging, such as MRI or CT scans of a patient's own anatomy. This allows for the creation of highly personalized and anatomically accurate structures. The bioprinter works much like a conventional 3D printer, but with biological precision. It extrudes the bioink layer by layer, building up complex architectures. These initial structures are often fragile and require further development. Once printed, these cellular constructs are placed in bioreactors. These specialized environments mimic the conditions inside the human body, providing nutrients and mechanical stimulation. This allows the cells to mature, differentiate, and organize into functional tissues. Over time, the cells proliferate and secrete their own extracellular matrix. This natural scaffolding replaces the initial bioink polymers, resulting in a fully biological, living tissue. The goal is to create structures that can integrate seamlessly into the body. Significant progress has already been made in bioprinting simpler tissues. Researchers have successfully printed functional skin grafts, cartilage, and even bone structures. These advancements are already moving towards clinical trials for various applications. A critical milestone has been the successful printing of vascular networks. Blood vessels are essential for delivering oxygen and nutrients to cells within any organ. Without a functional vascular system, larger printed tissues cannot survive or thrive. While printing a fully functional complex organ like a heart or kidney remains a formidable challenge, the foundational steps are firmly in place. The ability to create intricate microarchitectures is paving the way for more sophisticated constructs. Despite the incredible progress, several significant hurdles remain. Vascularization, the creation of a dense and functional network of blood vessels throughout a printed organ, is perhaps the most critical. Without it, cells deep within the structure will quickly die. The sheer complexity of human organs also presents a challenge. Organs are not just collections of cells, they are intricate systems with multiple cell types, precise spatial arrangements, and complex signaling pathways. Replicating this complexity is a monumental task. Regulatory approval and ethical considerations are also paramount. Ensuring the safety, efficacy, and long-term viability of bioprinted organs requires rigorous testing and a clear regulatory framework. Public acceptance and ethical guidelines must also be carefully considered. The potential impact of 3D bioprinting on healthcare is immense. It could revolutionize drug discovery and testing by providing accurate human tissue models. This would reduce reliance on animal testing and accelerate the development of new therapies. Personalized medicine stands to benefit enormously. Organs could be printed using a patient's own cells, virtually eliminating the risk of immune rejection. This would remove the need for lifelong immunosuppressive drugs, improving patient outcomes and quality of life. Ultimately, 3D bioprinting holds the promise of ending the organ shortage crisis. It could provide an unlimited supply of functional organs, transforming the lives of millions and offering a new lease on life for those currently waiting. The journey to fully functional bioprinted organs is a long one, but the trajectory is clear. Continued investment in research and development is crucial. This includes funding for basic science, advanced engineering, and clinical translation. Collaboration across disciplines, biology, engineering, medicine, and materials science is essential. By pooling expertise and resources, we can accelerate the pace of discovery and overcome the remaining technical challenges. The vision of manufacturing human organs is no longer a distant dream. It is a rapidly advancing field poised to redefine healthcare. By supporting this transformative technology, we are investing in a future where organ failure is no longer a death sentence, but a treatable condition.